Good afternoon, everybody. Whether my voice is audible?
Yes, it is audible, sir.
Yes, sir. Thank you, sir. Thank you for confirmation. Uh, Good afternoon, esteemed guests, registered participants, and distinguished colleagues. As the hush descends and the spotlight narrows, it amplifies the magnitude of the occasion we are here to witness. So, I am Dr. C.H. Praveen Kumar, Vice Principal of Siskandreddy College of Pharmacy, and your host for this live webinar today. And I extend a heartwarming welcome to each and every one of you to the one-day webinar on building blocks of pharmacology, the essential components of anatomy and physiology in comprehensive common sense. We are happy to have among us our highly respected and esteemed presence, our chief guest, Mrs. I. Aparna Lakshmi. Thank you, ma'am, for giving this opportunity for esteemed institution. So we invite our secretary, Sri K. Siskant Reddy Garu, president of LES Sri K. Radhika Garu. And thank you so much, sir and madam, for joining us in this afternoon. I invite our principal, Dr. V. Harivaskar sir, and my colleagues and the participants online as well as YouTube Live for participating in this live webinar. Thank you. Light is the symbol of brightness and prosperity. As light expels the darkness, and similarly, blessings bring in our life prosperity and happiness. To make this afternoon a blessed one, invoke God in Vinayaka by kindling the lamp of knowledge and wisdom.
दैवताय गणाध्यक्षाय धीमहि गुणशरीराय गुणमण्डिताय गुणेशानाय धीमहि गुणातीताय गुणाधीशाय गुणप्रविष्टाय धीमहि एकदन्ताय वक्रतुण्डाय गौरीतनयाय धीमहि गजेशानाय भालचन्द्राय श्रीगणेशाय धीमहि एकदन्ताय वक्रतुण्डाय गौरीतनयाय धीमहि गजेशानाय भालचन्द्राय श्रीगणेशाय धीमहि.
So thank you all for your patience and blessings from Lord Vinayaka. So, sir, good afternoon, sir. Principal, sir.
Good afternoon, sir.
Sir,
Yes, sir. Good afternoon, sir. Tell me, sir.
so let me introduce a brief introduction about our college first, sir. So afterwards we move on to the
Yes.
concept actually. So let
Please,
me
sir.
introduce our college in this way that Siskant Reddy College of Pharmacy was established in the year 2004 under Lindy Educational Society to impart quality education to our students. for their all-round development in order to cater the needs of the society and to achieve global excellence. So our vision is to the pinnacle to glory in pharmacy education is by elevating the institute to global level and also as a center for excellence. So it is achieved by a systematic and relentless approach to knowledge and the value-based practice in the profession. So we believe in being yourself, being unique, and stand out. And our mission is to embrace the pursuit of excellence both inside and outside the classroom. We encourage our students in critical thinking and emphasize the learning process to sow the seeds of discipline and professionalism among the faculty and students, providing state of art infrastructural facilities and latest innovative methods. So now I would like to play an introductory video of our college. So thank you for viewing the video. Now I would like our principal to introduce our chief guest briefly. Sir,
Yes. Good afternoon. Can I start, sir?
yes, sir, please.
Uh, Good afternoon to everyone. On behalf of Sasikant Reddy College of Pharmacy, I feel honored to welcome our Mr. and Mrs. Chairman, today's chief guest, Mrs. I. Aparna Lakshmi, Associate Professor, NAT Pharmacy College, Raichur, Karnataka. All the attendees, yes, in fact, they are the backbone of success for this event, and my fellow colleagues for taking a keen part in this important program. See, today's chief guest, our chief guest, respected madam, I, Aparna Lakshmi, On behalf of Sasikant Reddy, I welcome you, madam. Since 2010, I know about her. Yes, she is very sincere, like a pharmacist, teaching faculty. I am so proud on that. Today, our Vice Principal, Dr. Praveen Kumar, arranged this webinar. We started the first webinar, I think. So, in that webinar also, she is coming. So, thank you once again, madam. In the busy schedule, within the busy schedule, you are accepting our offer. So, the topic, webinar topic is comprehensive drug understanding. See, this predicting and understanding comprehensive drug is always a tricky one. So many things are there in this topic. So here, so many drug-drug interaction, adverse reactions, soma effect, physiology, anatomy, physiology. So many factors involves in this comprehensive drug understanding. Being a pharmacist, it is far most important. It is in a syllabus or not, that is different. Being a good pharmacist, you must have the See, it is, for example, drug interaction, it is always cause unexpected and uneven adverse drug reactions. So that it is important to identify drug interactions before the drugs are used in the market. However, principle the prediction I mean what I mean prediction or identification of drug interaction requires much more time. Moreover, lot of money we have to spend on that. So for that reason, as a comprehensive way, we are taking computational approaches we are doing. So like that we can overcome one by one and understand. It is very helpful for understanding our comprehensive drug. So here, chief guest, Mrs. So here, sir, can you take a point?
అయిపోయింది మేడం, అది అయిపోయింది. అయిపోయింది మేడం, ఇది కొట్టేసాను.
Basically, she's a pharmacologist.
ఫస్ట్లోనే ఫస్ట్ ఆప్షన్ అడ్వాన్స్డ్ డీటెయిల్స్ ఫస్ట్లోనే ఉంటుంది.
Pharmacology.
మేడం,
So
నేను
in
యాక్చువల్గా వెబినార్
in
ఉన్నాను,
that
స్పీకర్ ఉంది,
she
దానికోసం
may
ఒక
explain
2
you
minutes నేను కాల్ చేస్తాను మీకు. నేను
anatomy,
చేస్తాను.
physiology,
అదే
con-
ఫస్ట్ దాంట్లోనే ఉంటుంది మేడం, ఫస్ట్
uh,
ఫస్ట్ దాంట్లోనే. మన కింద మీకు
system, autonomic nervous
పీకే అంటే ఎలక్ట్రికల్ ఎలక్ట్రోమాగ్నెటిక్, తప్ప పీకే రాకూడదు. అది ఎస్ కొట్టండి,
all
అది ఎస్ కొట్టండి, అది ఎస్ కొట్టిన
once
తర్వాత
again
అంటే
to the webinar and
రికార్డింగ్
hope you will time ahead
ఓకేనా, కొడితే అప్పుడు మీకు
and all.
బ్రేక్ తీసుకుంటున్నాను. ఓకే మేడం, నేను మళ్ళీ
Thank
చేస్తాను
you.
5 minutes.
And Dr. Praveen Kumar, you can take over. Hello, sir.
థ్యాంక్ యూ సార్, థ్యాంక్ యూ
Is it
ఫర్
audible?
గివింగ్ వెరీ బ్రీఫ్ ఇంట్రొడక్షన్ అబౌట్ ద టాపిక్.
Hello.
ఈజ్ ఇట్ ఆడిబుల్ సార్?
Yes, yes, completely audible.
Yeah, sir. Yes, sir. Yes. Now I would like to introduce our speaker with the help of our PowerPoint, sir. Sir, whether PowerPoint is visible, sir?
Yes, yes, visible, sir.
Yeah, sir.
Visible, sir.
Let me introduce briefly about our chief guest today, Mrs. I. Aparna Lakshmi. She has studied her BPharm from Rouse College of Pharmacy, which is situated in Nellore in 2008. She has completed her BPharmacy in 2008, and she has completed her MPharmacy in the Department of Pharmacology in SRM College of Pharmacy, comes under SRM University, Chennai, and she have achieved gold medal in PG in 2010. Coming to her career, she worked as an assistant professor in the Assam Institute of Pharmacy from May 2010 to February 2013, and she has worked as an assistant clinical coordinator, Excel Health Bangalore, from March 2013 to November 2013, and she worked as an assistant professor in Oxford College of Pharmacy, Bangalore, from RGUHS November 2013, November 2013 to August 2016, and she worked as an associate professor at Raghu College of Pharmacy from July 2018 to October 2021, and presently now she is working as an associate professor in NET Pharmacy College, Raichur, Karnataka, from 2022 to till date. Coming to her achievements, she is an approved PG guide in the Department of Pharmacology from Rajiv Gandhi University of Health Sciences, Bangalore. She is approved PG examiner in the Department of Pharmacology from Rajiv Gandhi University, and she has served as a question paper setter, and she has been as an evaluator for many universities, and finally she has published at more than 25 national and international journals. Once again, I invite Mrs. Aparna Lakshmi. Thank you, madam, for giving this opportunity for us to share your views for the students of our college. Aparna, madam.
Sir, am I audible, sir?
Yeah, yes.
Yes, it is audible.
Madam, shall I share the PowerPoint, madam? Aparna, madam? Aparna, madam?
Aparna, madam.
Am I audible?
Audible, Aparna? Aparna, your voice is not audible, please.
Sir, am I audible now, sir?
Yeah, yes.
Am I audible?
Yes, it is. Can I share the PowerPoint, madam?
Yes, sir, sure, sir.
Yeah, thank you, madam. Thank you. Whether can you see the PowerPoint, sir? Madam?
Yes, sir, yes.
Yeah,
Yes, sir, yes,
yeah,
sir.
please continue.
Yes.
Thank you.
Can I start, sir?
Yeah, yes, madam. Thank you.
Yes, of course.
Good afternoon to one and all. Before starting the session, I would like to thank Secretary Dr. Seshikant Reddy, sir, and Chairman ma'am for giving me an opportunity to be as chief guest and resource person for today's session. And I would also like to thank Dr. Haribhaskar sir, Principal, and Dr. Praveen sir, Vice Principal, Shashikant Reddy College of Pharmacy, for giving me a chance to be a speaker today. And today we are going to discuss about building blocks of pharmacology, the essential role of human anatomy and physiology in comprehensive drug understanding. Good afternoon to all the students. Am I audible?
Yes, madam. It is audible.
Perfect.
Right. So let us have an interactive session along with this. So by seeing the topic heading, don't feel that it is very comprehensive thing. It's a very easy one. So what I'm trying to say is in this session, what is the use of reading HAP and how we can apply it in understanding the mechanism of drugs as well as while reading the pharmacology of other drugs. Usually when we read HAP, we read about various systems and we read just anatomy and physiology and we'll forget it in the first sem, right? So when we come to pharmacology in fourth sem, we feel little difficulty because we don't apply whatever we are reading in HAP there, right? So let us start with the session. Sir, can we go to next slide, sir?
Yes, sir. Madam, second slide, it's not visible.
Not visible, sir. Yes, sir. Yes, sir. Right. So coming to anatomy, anatomy is a branch of science which deals with study of structure of various parts of our body as well as structure of various systems and how they are interlinked with each other. Simultaneously, we also read about physiology, which is a branch of science which deals with functions of various bodies and body systems and their relationship with each other. For example, if we are reading about nervous system, we just read about its structures, various structures present in nervous system like brain, and also we also read about peripheral nervous system and their functions, right? So we don't correlate the functions of nervous system with other systems. For example, if you consider endocrine system, let us take pancreas. Pancreas secretes insulin and glucagon. We all know that. How insulin is released from pancreas? It is always linked with the functioning of nervous system. When the glucose levels are increased in our body, simultaneously the nervous system activates the pancreas to release insulin. So like this, each and every system is connected in our body and work together to maintain homeostasis in our body, right? And the structure of organs or the structure of systems vary from location to location depending upon the function of the area, that particular area. Sir, can we go to next slide, sir?
Okay, ma'am.
Sir, I can't move to next slide.
Yeah. Now,
Right. Can you see here, ma'am? We just take an example of skeletal system here. How many bones are there in the skull? There are 22 bones in the skull, 8 cranial bones and 14 facial bones. Is there any gap in the alignment of these bones? Can anyone answer? Is there any gap in the alignment of these bones? Are they arranged very tightly? Right. So what happens here is the bones are very tightly closed because the skull has to protect the brain, which is an essential organ for our body. So let us come to the bones present in the hand. So in the hand, the bones are not tightly closed. There are gaps between the bones. Why we have gaps between the bones in the hand? Because movements has to be allowed. We should be able to move our hand. So the bones are not attached here, whereas in the skull, the bones are closely attached. So much difference is there even though the bones belongs to the same system. So the alignment, the function varies from location to location, right? Sir, next slide, sir. Yeah. So let us have another example which helps us to understand about the organization of epithelial tissue. So imagine all of you would have studied about various tissues like epithelial tissues and types of epithelial tissues also. So here I have taken example of alveoli in the lungs as well as urinary bladder in the urinary system. So if you see the thickness of the alveoli, all of us know that alveoli helps in the exchange of gases. The thickness is very less here. Alveoli is so thin and it is organized in such a way that easily it can allow the exchange of gases. Whereas urinary bladder, the thickness of urinary bladder is more than all other organs because it has to store urine. It has to store urine and it has to allow stretching also because the normal urinary bladder capacity is from 400 to 650 ml, right? So there is so much difference between the arrangement of epithelium in alveoli as well as the arrangement of epithelium in the urinary bladder. Even though those both belong to the same tissue, still the arrangement varies because the function is different in different locations. In alveoli, the function is to exchange the gases, whereas in urinary bladder, the function is to store the urine, right? So these two examples makes us to understand that the structure function of organs vary from organ to organ or from system to system, right? So let us take one more example how anatomy helps us in understanding the diseases. Sir, next slide, sir.
Yes.
Yeah, right. I think all of you are able to see this diagram. This diagram divides the entire abdomen into 6 areas. Actually, there is a left hypochondrion, right hypochondrion, epigastric region, like this different areas are there. Usually we explain this before starting the digestive system. So how we can divide the abdomen into various sections. What happens here if we don't apply anatomy while understanding the diseases? Imagine a patient went to a doctor saying that I have pain in the left side of the abdomen. So the doctor can easily analyze where is the pain, what organs are involved here. What organs are there on the left side, ma? You can see there, left epichondrial region. You can see there is a part of stomach and there is a spleen too and a portion of pancreas, right? So easily a doctor can assess that there is some abnormality either in the stomach, pancreas or spleen. But being a pharmacist, we don't apply these. Just we read what are the organs of digestive system and what are their functions, right? So simply by saying the location, doctor was able to identify it. So we should also keep and analyze this data so that we can also be able to assess what is the problem according to the location, according to the symptoms explained by the patient, right? So similarly, you can say epigastric region. If a patient complains that I have pain in the epigastric region, you can say that he has some abnormality with the stomach. It is very easy to assess according to the position explained here, or a patient comes with some symptoms explaining that I have some problem in certain area, right? So this is also one more example which helps us to understand what is the applicability of HAP in understanding diseases, right? So and here I want to give one more example regarding prefixes and suffixes. I hope everybody uses Ross and Wilson or either Tartura for studying HAP. The very first day your staff would have told you to read prefixes and suffixes, right? Here, what is the importance of prefixes and suffixes? We may neglect it while reading HAP, but it is more useful when we read about diseases or to understand the pathophysiology of diseases. Here, I have come with an example of myocarditis. Have a look on that. Here, the suffix is itis. We all know that itis means inflammation or swelling at an area. Here, what area? What is the prefix presiding here? Myocarditis, myocard. So, myocard represents what? Myocardium of the heart. So, myocarditis, inflammation of myocardium. Very easy to assess. We don't want any explanation for this also. Easily we can understand if we read prefixes and suffixes in the first sem itself, right? So, we know it is very common. We'll read it. Let us take one more hard example. Cholestasis. We neglect, we don't read what is coli, what is status, because itis is very common, like hepatitis, nephritis, easy to analyze and apply. So here, coming to cholestasis, coli always represents bile and stasis represents stagnosis. I mean, there is no movement of fluids there, and the fluid has accumulated at a certain area. So combine the prefix and suffix here, cholestasis, that is bile was struck and there is no movement of bile from one area to another area. So if you recall the connectivity between liver, bile and intestine, we will go back to enterohepatic circulation, where the liver synthesizes the bile and passes through bile ducts, and it was stored in the gallbladder, and the gallbladder again releases it into the intestine. So here cholestasis means bile was unable to move from one area to another area, maybe because of tumor or maybe because of stones, right? So here just a prefix and suffix. If we remember the meanings of prefixes and suffixes from HAP, it is very easy to understand the basics of a disease like cholestasis. So with these examples, I think I have given a brief introduction about how HAP is useful in understanding the basics of a disease, right? And one more application of HAP. Sir, can we move to next slide, sir? Right. So the another application of HAP is to know parameters of normal health. To know parameters of normal health. Right. So I think all of you know about vital signs. What are the vital signs? Which are commonly checked when we go to a hospital. So in the hospital, they will check body's temperature, and they also check pulse, which helps us to know about heart rate and also respiratory rate, saturation levels of oxygen, that is blood oxygen, as well as blood pressure, right? While we read about the diseases, nowhere we repeat what are the normal values or what are vital signs. So we all read this from HAP. And vital signs, whatever disease you go to the hospital, first they check the vital signs. If there is any abnormality in the vital signs, then according to that, they go for further diagnosis of the disease. So what are the normal values of all this? We have to read from the HAP, and we should also apply them to the analysis of diseases. For example, let us take a small thing, blood pressure. What is the hypertension definition? Can anyone, or when we call hypertension, it is hypertension. What is the normal level of blood pressure? Can anyone answer? Right, fine. So if the blood pressure is more than 140, systolic blood pressure, and if the diastolic pressure is more than
stopping.
Yes.
Student, if they want to interact with the guest, you just unmute yourself and you can directly speak with them. No problem. You just unmute yourself and just directly speak or interact with the speaker. Thank you, madam. Please go ahead.
Yes, sir. Yes, sir. Right, ma? So here the blood pressure value, normal value is 120 by 80. What is 120? What is 80? What is systole? What is diastole? All we read it in HAP. For example, if hypertension is 140 by 90, still we consider it as hypertension because there was rise in the value of systolic blood pressure, right? So all these basics we have to read from HAP and we should apply it. Or if a patient comes to a hospital, these are the main things first we have to check. Sir, can we move to next slide, sir? Right. So what is the other applications of HAP? We are moving to next application. Factors affecting various physiological process and its effects, right? HAP helps us studying anatomy and physiology helps us to analyze various effects and various factors that change the physiological processes or its effects. I will not make it so complicated. So I have taken a small example here. See the image once what I have kept there. A person was climbing a hill. So what happens when a person climbs a hill? What happens to their respiratory rate? What happens to their blood pressure? I have not taken an example of any disease. Normal physiological process. In a normal physiology, if a person is climbing a hill, what happens to their respiratory rate? Either it increases or decreases. Anybody?
Decreases.
No, it has to increase because when we are climbing a hill, automatically the requirement of oxygen, the requirement of blood supply should be more so that it can meet the demands of our body. So obviously the respiratory rate increases to meet the demand of the body, right? So here, this is not a disease, no? This is just body needs a higher requirement. So our body will adjust. It will, the CNS system sends impulses to the lungs so that simultaneously the respiratory rate can increase and we will be able to supply oxygen to the body properly, right? And here there is one more thing, but still I will try to simplify it. There is one more system called respiratory drive. Respiratory drive. In CNS, respiratory center is located, and this respiratory center can control the respiratory rate. So obviously, when oxygen levels come down and when CO2 levels increase, so when a person is climbing a hill, if the altitude is increasing, the CO2 levels will go up and the oxygen levels will come down. Immediately, there are some receptors which can sense these levels in the blood, and the impulses are sent to the central nervous system, which will try to control the respiratory rate so that the respiratory rate increases and respiration will be fast and it will try to supply more oxygen to the body, right? So like this, HAP helps us to analyze the situation, and it also helps us to know about the factors which will vary the physiological process. Here I am stressing out, it is physiological process, it's not pathological process. I am not stressing out about pathological process, right? So I can give one more example. Suppose a person is standing for a long time, suppose like 1 hour or 2 hours, and they sat at once. Or you can also take early in the morning after having a good sleep, when we try to get up, there will be change in our body position. So obviously there will be change in blood pressure because of change in body position. So are we falling down because of low BP early in the morning? Anybody? Because of change in body's position? Do we have any fall in blood pressure? Do we have any syncope? No, right? No, right? So, because our body was able to maintain it, and we have many mechanisms which can control the changes during a physiological process, right? So to make it so simple, I have taken one more example here. Sir, can we move to next slide, sir? If students have any doubts, you can ask me even between the session also. Yeah. So before explaining one more example, what is homeostasis? This is also a basic thing which we read during introduction of HAP. Can anybody know what is homeostasis?
Same state.
Yeah, very good, very good. Homeostasis is nothing but maintaining the stability of our body because of a change in external environment. By seeing the definition, it is common as a student to feel that the topic is very difficult. Let us skip it or let us leave it, right? So it is very simple, homeostasis, maintaining the balance in the body because of change in external environment, right? So here, let us imagine we are standing in a cold temperature. We are standing in an area where the temperature is very low. What is our normal body temperature? Anybody? Normal body temperature? Fine. So normal body temperature, yeah. The normal body temperature is 37 degrees centigrade. So when we stand in a cold area, is our body temperature going down? No. Do we feel any coldness like chill and shivering? Yes, obviously. So why the shivering happens? Why we feel too cold is to maintain body temperature. Again, central nervous system sends impulses to the skeletal muscles. The muscle starts contracting repeatedly. Because of that, we feel chills. The repeated muscle contractions produces heat in our body so that our body temperature is maintained at 37 degrees centigrade without change in it, right? This is not a disease, ma. Again, this is normal physiological process. That is, our body parameters are always maintained at a normal level because of this homeostasis, right? And this homeostasis works with two mechanisms, positive feedback and negative feedback, right? So what is negative feedback? Negative feedback is if anything is opposite, it will bring it back to the normal. Very simple. Negative means always opposite, right? So if anything is opposite, for example, body temperature normal, body temperature is decreased. What is opposite to it? It has to be increased. So our body system will try to increase it and tries to bring back to the normal, right? So here I have taken one more example of blood pressure. So how blood pressure is maintained in the body within the normal limits. So here I have taken an example of negative feedback mechanism. What are all the components present in feedback mechanism? There will be a stimulus. Stimulus is nothing but change in the parameter. Let us take blood pressure, blood volume, or body temperature. It might be anything. And then sensor. Sensor means which can sense the change in it. Suppose change in blood temperature, change in body, blood pressure, like that. And then control. What is the control organ? Control organ is brain in our body. And what is effector? What brain can do to correct that change in stimulus, right? So let us go to this diagram here. The last one, left corner. So some stimulus disrupts homeostasis. That is, here there is increase in blood pressure. Increase in blood pressure, right? So which can sense this change in blood pressure? I hope all of you know about baroreceptors, right? What are baroreceptors? Where they are located? Anyone? Baroreceptors, where they are located?
Brain.
Okay, fine. But these baroreceptors, actually, they are located on the aorta, major aorta, which arises from left ventricle, right? So from left ventricle, these receptors, these are just like, imagine like our CCTVs. CCTVs captures everything, no? So same like that. These receptors can sense change in blood pressure. Slight change in blood pressure also can be sensed by this. And immediately they send that impulses to the brain. There is change in blood pressure. You have to control it. So our brain is like CPU to the system, like that. So it will take the input and immediately, accordingly, it will act. I will explain you in the last slide how this is controlled with the help of sympathetic and parasympathetic system. So here I am just briefing it out. So here it helps us to understand how a system is controlled with this homeostasis mechanisms in the body, right? So when I go to last slide, I will explain you how this is applicable. Okay. So with this, I have given a brief introduction of HAP and its applications. And now we are moving to application of HAP in studying pharmacology. So before going into that, so let us have a look on what is pharmacology, what is a drug. Then I will move to it more because many freshers also might be there or first year students also might be there. So let us know what is pharmacology or what is a drug, right? So if any fourth sem students are there, can anyone can respond? What is pharmacology? Anybody from fourth sem or fifth sem? Okay, fine. So what is pharma? Yes, ma? Okay. So what is pharmacology? Here, pharmakon is a word derived from Greek, which says drug. And ology means study. Wherever you get ology is usually deals with study. So in a simple language, pharmacology is nothing but study of drugs, right? So what kind of studies we do? What kind of studies we read under pharmacology regarding a drug, right? So before going to this, what is drug? What is drug, ma? Can anybody define what is drug? Can anybody define what is drug?
Medicine.
Yeah, Ganesh, yes, right. So, what is drug? Actually, it has two definitions. When we ask students and Viva, usually what they answer they give for drug. They say it is a substance which used to treat a disease. That's all. One line answer we give usually. So, which is used to treat a disease. It's not like that. Anything which is used, any substance, any chemical moiety, let it be a plant derivative or any extract derived from plant or any other substance like what compounds were used in homeopathic medicine or Ayurvedic medicine, whatever it is, any chemical entity which is used for diagnosis, prevention, treatment, or cure of a disease, right, ma? So here, fine, treatment, cure means we know. What about diagnosis and prevention? Right, no? So vaccines, what are vaccines? Can we call vaccines as drugs or we can take whenever we get infection? What for vaccines are used, ma? Anybody? What are vaccines? Yes, vaccines were used for prevention. Before getting the disease itself, we take vaccines as a preventive measure so that we don't get that infection, right? So that vaccines also fall under the category of drugs, right? So here, okay, fine, this definition is okay, but we have some exceptions. What about oral contraceptives, which are not used to treat any disease? And what about multivitamins, which we take regularly? Those are also not used to treat any disease or to diagnose or to prevent something, right? So how can we change the definition of drug, right? So here, this definition was changed by WHO and it is defined as drug is any substance or product that is used or intended to be used to modify any physiological system. Here, no, we are not using a word called treatment or a word called prevention. Here we have modified the definition and drugs are defined to modify physiological systems or pathological state. That is, what for we take vitamins to make us healthy, right? Fine. So, we are trying to improve our health status, right? What for oral contraceptives are used? Oral contraceptives are used to prevent fertilization of egg in females, right? So there also it is not used for a disease. We are altering a process physiologically, right? So that's how the definition is changed and is applicable to any moiety either which alters the pathological state. Pathological state means disease, which is either used for diagnosis, prevention or treatment of a disease or physiological systems too, right? So with this, I think I have given brief introduction about what is pharmacology and what is drug, right? So let us go to next slide. Sir, can we move to next slide, sir? Yeah. So coming to pharmacokinetics. Pharmacology is a branch which actually deals with pharmacokinetics and pharmacodynamics. What is pharmacokinetics? Pharmacokinetics means what happens to drug in our body. Right, ma? I hope all of you can understand in Telugu too. To make it simplify, because many of them might be from first year. మనం ఒక టాబ్లెట్ తీసుకుంటే టాబ్లెట్కి ఏమవుతుంది, మన బాడీలో టాబ్లెట్కి ఏమవుతుంది, రైట్ మా? సో ఫస్ట్ ఇట్ విల్ బి ఎస్ మా, ఎస్ రైట్ మా, రైట్. సో pharmacokinetics usually deals with absorption, distribution, metabolism and kinetics. Okay, so let us start with absorption. What is absorption? When we take a tablet, first it gets solubilized, that is it converts into a small particles like fine particles from stomach. Usually we take many drugs through oral route, so from stomach these particles slowly get entered into blood through many absorption process, transport process like active transport, passive transport, depending upon the nature of the drug. I'm not going to complicate it much. What I mean to say is, in absorption, we study about entry of drug molecule from the site of administration to the blood. From the site of administration. Administration means let it might be oral route or intravenous route like injections and many other routes. From any site, that is from muscle. If a drug is injected to the muscle, from muscle it has to go to the blood. If it is given subcutaneously, that is under the skin, from skin it has to go to the blood, right? Anybody has doubts, ma? Fine. So here, in absorption, how HAP helps us. So in the very first chapter, we study about cell membrane. Yes, no, ma? We study about cell membrane structure, and various transport mechanisms like active transport, passive transport, all this, right? And here I have explained you how a drug, what happens to drug from the site of administration, right? So all drugs can be easily moved from blood to all the parts of the body, but the problem comes with CNS. Why? Why is the problem with the CNS? What happens there? Can all the drug enter into CNS, or is there any restriction? Sir, can we move to next slide, sir? Yeah, ma. So see here how what is the barrier between before going to explain this? Everybody knows about blood-brain barrier. Blood-brain barrier and its importance, how it is formed. Anybody? Right, fine. So here, the blood-brain barrier, the name itself explains us blood-brain barrier. That is, the barrier is in between blood and brain.
Ma'am,
Yeah.
this is selective permeable membrane between the blood and the intestine of the brain, ma'am.
Yeah, very good, very good. Here, the barrier is formed between blood and brain. And here the barrier is formed by astrocytes. Astrocytes, you can see the organization. So how these cells, the pedicles, that is leg-shaped pedicles, are attached to one another without allowing any gap between the cells. So this alignment does not allow all the substances to cross through the barrier and enter into the central nervous system. This is how our brain is protected. Then what kind of drugs can cross this barrier? Lipophilic drugs. Lipophilic drugs means which are more soluble in lipids, or which is lipophilic, right? So these kind of drugs only can cross this barrier and reach the central nervous system. So when we will be able to understand, suppose you are reading about drugs used in central nervous system. So they don't explain all this there. Directly they say the drug is lipophilic. So you have to understand that these drugs are lipophilic. They easily cross blood-brain barrier. So they are acting on the brain. Right, ma? So this is the applicability when we read about blood-brain barrier in HAP. It makes us to understand how the drugs are able to cross and reach the brain and show the action. Right, ma? Sir, can we move to next slide, sir? Yeah. So the next heading under pharmacokinetics is distribution. What is distribution? Now, once if a drug molecule reaches blood, what happens to the drug?
Excuse me, ma'am.
Yes, ma'am?
Ma'am, what is the definition of pharmacodynamic and pharmacokinetic, ma'am?
Yes, ma'am. Pharmacokinetics. I am going through kinetics. Pharmacodynamics I'll explain in the upcoming slides. Pharmacokinetics means study of or branch which deals with what happens to drug inside the body. What happens to drug inside the body, or a branch which deals with drug, what happens to drug inside the body. Can we move to first slide? It was mentioned there.
Yes, ma'am.
Yeah. So I will simplify it, Ganesh. Kinetics means what, ma? Kinetics means what? Kinetics is generally movement, right? No. So similarly, what happens to the drug and how it moves from one area to another area in the body, right, ma? And kinetics have several headings like absorption, distribution, metabolism, and excretion. I am explaining one by one, and how HAP is useful in all this to understand about the pharmacokinetics of drugs. Right, Ganesh?
Yes, ma'am. Okay, ma'am.
Right. So we are moving to distribution, ma. So coming to distribution, Generally, distribution means what? Anybody? Distribution?
Drugs inside the body, how to distribute throughout the body,
Okay.
and
Yeah,
after
yeah.
that they lead to metabolism.
Right. So see here, once look at the slide. We have learnt about absorption. Absorption means movement of the drug from the site of administration to the blood, right? So once when the drug reaches the blood, how it is distributed throughout the body? So it is distributed because the blood circulates through systemic circulation. The blood reaches all the organs. So that's how the drug also reaches all the organs in our body. Here, while reading distribution, we should know about the plasma proteins and their influence in the body. I hope all of you would have read about composition of blood and plasma in HAP, right, ma? So when we read about composition of blood, we write about plasma proteins, components of plasma, and we also read about formed elements. Formed elements means RBC, WBC, platelets. We don't have any issue with them. So here issue comes with plasma proteins. What are plasma proteins? I think many of you would have read in HAP, albumin and globulin. These are plasma proteins. So in HAP, we stop with that. We don't actually read anything more than that. But more explanation has to be given about albumin and globulin there. Because that makes us to understand this pharmacological part. Right, ma? What happens here is many drugs binds to albumin. Many drugs binds to albumin. Albumin is a plasma protein. If any drug binds to plasma proteins, right, ma? So that drugs cannot show action because they will be in bound form. They are not in free form. Drug will show action only if it is in free form. Then only it can go and bind to any receptors and show action. Right, ma? So we'll not complex it. We'll not make it too complicated. So I want to say that free, if the drug is in free form, then only it can show action. If it is in bound form, it can't show action. Right, ma? So these plasma proteins binds to the drugs and they convert this drug into a bound form. Right, ma? So this is one of the disadvantages, but there are many advantages with plasma protein binding. So what are the advantages? Drug stays in the body for longer duration. If the drug stays in the body for longer duration, slowly the drug molecules will be released into the blood and they will also show the action. So the duration of action will be for longer time. For example, if a drug is acting for 6 hours, if that drug has plasma protein binding, it can act for longer duration, 1 hour or 2 hours extra like that. Fine, ma? So here we have both advantages and disadvantages with plasma protein binding. And if you read that in HAP, so what are albumins, what are globulins, do they have really binding sites and all? It becomes easy to understand about that when we come to HAP. Fine, ma? So can we move to next topic? Sir, next slide, sir. Yeah. So what is metabolism? So this slide will be very interesting, ma. If anybody is sleeping, all of you will be activated now. See, metabolism of liver. Metabolism in liver. Now, whatever drugs or whatever substances which enters into our blood, all the substances are detoxified in liver. That is, once after showing the action, the drugs will be metabolized in the liver. They are converted to less toxic form and easily excreted out of our body. So here, to make it very simple, I am taking the example of alcohol. So what happens to alcohol after entering into blood, right? So alcohol, let it be 1 ml, 2 ml, 10 ml, how much ever we consume, entire thing goes to liver for metabolism. You can see the diagram, ethanol metabolism. Ethanol is converted to acetaldehyde. Acetaldehyde is converted to acetate and acetate is converted to acetyl-CoA. I hope if the students are in second sem, they will also read about biochemistry and they would have read this under liver function tests and they would have read about acetyl-CoA under citric acid cycle. Right, Nama? So here, what is the problem, ma'am? Ethanol is getting detoxified. No, ethanol is getting metabolism. No, what is the problem? So we all know that if a person is consuming alcohol, the first organ to be damaged is liver. Why? Why liver means? I told Nama, even if it is 1 ml, 2 ml, the liver detoxifies the alcohol. So what happens here? The first product in ethanol metabolism is acetaldehyde. Is acetaldehyde that toxic? Yes, acetaldehyde is toxic. This acetaldehyde damages the liver. Let it be whatever the concentration is, it damages the liver. So initially, first day they consumed some 100 ml, second day 200 ml, third day. So if the quantity is going on increasing, the extent of liver damage also increases, right? So usually we'll have a question, what is the safe dose of alcohol? There is no question, it is zero. There is no safe dose of alcohol. Whatever we consume, it is toxic to the liver. Fine, ma? So here, the end product, acetyl-CoA, again it goes to citric acid cycle for energy production. So this is also a burden to the liver again. Right? This is also a burden. Already it is in stage of damage and that also becomes a burden for the liver. Right? So here there is one more I have given. One more example, ma. So can you see here the diagram of heart? The diagram of heart. Yeah. So how this heart is related to metabolism, right? So here I'll give a small example, ma, how HAP is useful for us. Maybe under the functions of liver in the digestive system, you would have read about liver is a detoxification organ. It stores many things. It helps in synthesis of vitamin K. So many functions you would have read just for heading sake, and we will write it off. Fine. So here liver produces two enzymes, SGOT or SGPT or ALT or AST. If you are reading pathophysiology, definitely you will know about this. So these two enzymes are secreted by liver, and if their levels are very high, we can say that the person is suffering from a liver disease. Then what is the link with heart here means? I'll take an example here just for fun, to make you understand. Before marriage, suppose if a boy wants to get married, we check their financial status, we check many other things. Yes, right, no? So instead of checking all this, you can ask the person to go for one blood test, single blood test. What blood test? SGPT blood test. And what is this SGPT? My SGPT is nothing but serum glutamate pyruvate transaminase. Serum glutamate pyruvate transaminase. What is this enzyme, ma'am? Why it is so important that we have to go for a blood test before a marriage, right? Imagine a person, a boy is around 26 or 25 years old. The SGPT levels should be below 10 or 20 like that. Suppose if the SGPT levels are 80, if the SGPT levels are 80 at the age of 26, definitely that person will have a chance of cardiac arrest at the age of 36. Definitely at the age of 36. Why, ma'am? This SGPT value indicates about inflammation in the body. Indicates about inflammation in the body. Do we read all this in HAP? No, we just read liver secretes two enzymes, serum glutamate pyruvate transaminase, that is ALT or AST. That's all. We don't read that. So we have to read its importance also. SGPT is very important and it tells about inflammatory levels in the body, especially liver inflammation and heart inflammation too. So that's why the single test, it just costs 80 rupees or 100 rupees, which says about our health status. Is there any inflammation of heart or liver in the body? So such importance is there for HAP, which helps us even to diagnose a disease or even to understand about enzymes and their role in pathophysiology of a disease. Right? Sir, can we move to next slide, sir? Yeah, ma. So the last topic under pharmacokinetics is excretion. I will not make you bored. I will make it so simple. I'll move fast. So coming to the excretion, we are not reading much about pharmacokinetics. Let us read about HAP. So in HAP, under urinary system, you would have read about steps involved in urine formation. What are the steps involved in urine formation? Anybody? Ganesh, what are the steps involved in urine formation?
Tubular secretion,
Yeah.
tubular
Yes.
filtration,
Yes.
absorption of water.
So now,
Absorption of water.
yes. So now,
Absorption of water.
yes. So now,
Absorption of water.
Yeah, there are three important steps in urine formation. Glomerular filtration. Ganesh, mute yourself, ma. Ganesh, mute yourself. Yeah. So glomerular filtration, tubular reabsorption, and tubular secretion. These three are important steps in urine formation. Glomerular filtration, that is the blood is getting filtered at the glomerulus. And next one, tubular reabsorption. Whatever filtered at the glomerulus will never be excreted, because if that much amount is excreted, will die in 1 hour easily. Why means what? The glomerular filtration rate, that is how much blood is filtered at the glomerulus. It is approximately 120 ml per minute. 120 ml per minute. If you can count it for 1 hour, it will be in liters. So we don't have that much blood volume. So we will easily die within 1 hour if this much amount is excreted from kidneys, right? So all that whatever is filtered at the glomerulus will not be excreted or will not be turned into urine. So here the next important step is tubular reabsorption. Reabsorption, whatever is getting filtered, again it goes back to the systemic circulation. That is, it goes back to the blood, like water, important ions like sodium, potassium, and then important constituents like glucose, amino acids, all those get reabsorbed back into the blood. And tubular secretion. So some of the ions also get excreted, that is secreted into the nephron. Some components can be secreted into the nephron. So all this finally get excreted through urine. Now how this is useful in pharmacology, or how it is helpful, and what we can learn here or apply from HAP. Now listen here, glomerular filtration rate 120 ml per minute. Now if the glomerular filtration rate is very less, for example 40 or 50, what does that indicate, ma'am? Anybody? If the glomerular filtration rate is very less, what does that indicate? Fine. So glomerulus is not doing its function because of some abnormality in the kidneys. So glomerular filtration rate helps us to analyze about kidney function. And next thing, when we are reading about drugs, we say these drugs are not filtered at the glomerulus, but secreted through tubular secretion. They are excreted out of the body through tubular secretion. Right, ma? For example, if two drugs were given simultaneously, which will excrete from the body through tubular secretion. I have given here a list of compounds which are excreted through tubular secretion. Acetazolamide, bile salts, hydrochlorothiazide, like that. Imagine two drugs were simultaneously taken by a patient, and both the drugs has to be secreted by tubular secretion. So the drugs will fight. I will go first. No, I will go first, like that. So only one drug can be excreted. The other drug cannot be excreted. So if the other drug cannot be excreted, that levels will increase, and that may lead to toxicity. When you will come to know all this, if you know about how the drug is excreted. If you know how the drugs is excreted, the doctor will not prescribe both drugs together. They won't prescribe because it may lead to toxicity. So this is how HAP is applied in excretion of drugs. Sir, can we move to next slide, sir? Yeah. So the last topic, here I want to talk about sympathetic and parasympathetic system or peripheral nervous system. So why I have taken this is many students in the first STEM, when we say about nervous system, by the name of the topic itself, they will come to a conclusion. Hey, no need to read this chapter. Better to skip it because we are not going to understand anything in nervous system. We'll leave it. Fine? Okay, fine. In HAP, you will leave. What about pharmacology? What about pathophysiology? You can't leave it because there are many diseases under central nervous system. Many drugs under central nervous system, which you cannot skip. So if you don't have basics in HAP, what you will read there? It will be very difficult for you. So just I am going to give a brief introduction about what is peripheral nervous system. Because CNS is very easy. We know that it has brain, it has spinal cord. So we'll read the functions of cerebrum, cerebellum. It becomes very easy for us. So toughest part is peripheral nervous system. What is this peripheral nervous system? It is so easy, ma. Before going to peripheral nervous system, I want to say about sensory nerves and motor nerves. Depending upon the function of the nerves, we divide the nerves into sensory nerves or motor nerves, right? Sensory nerves means which will collect information from all the other body parts and sends that information to the brain. Okay. And motor output or motor neurons means which will send impulses from the brain to other parts of the body. Okay. So if I say like this, nothing will go into our brain. I know that. So let us take an example, right? So you are walking on a road. Suddenly you saw a snake on the road. What you will do? Nobody is there next to you. What happens there? What you will do after seeing a snake? Anybody? You'll stand there. Yeah, you will not say hi to the snake, no? You will run away. So what is the sensory input here? What is the sensory input here? The image of the snake. From where it is going? From eyes. Whatever image we see or whatever object we see, that image falls onto the retina. Retina sends that impulse to the brain through optic nerve. Brain analyzes it. Hey, this is very dangerous. It may bite you. So what brain has to do? It will send motor impulses. How? It will send impulses to the legs, skeletal muscles. Run away fast. Otherwise you may die. So what is the sensory input here, ma? You are seeing a snake. Through eyes, input is going. What is the motor output? From brain, we are getting impulses to the skeletal muscles and immediately we are running away. Right? Very simple, no? Sensory input, motor output. Fine. So what all, you can see the image in the PPT. What all comes under sensory input? Somatic senses. Like if you touch, somatic senses means touch, like if you touch any hot object or movements. You have to walk on a narrow road or you are cycling. You have to maintain the balance, no? So all those comes under somatic senses and special senses. Special senses means vision, hearing, taste. That is special sense organs. All those comes under sensory input. Now coming to motor output. This motor division explains us about peripheral nervous system. Motor output has two again, somatic nervous system and autonomic nervous system. Very clear here, very easy to understand. What is autonomic nervous system? Autonomic nervous system includes the motor neurons. What are motor neurons, ma? Motor neurons means impulses, nerves which carry impulses from the brain to the other organs. So here autonomic nervous system means what organs? Smooth muscles, cardiac muscle and glands. Only this will come under autonomic nervous system. So it is very clear that autonomic nervous system mainly includes motor neurons which goes to smooth muscle, cardiac muscle and glands and GIT also. Right? It is very simple now. Here this autonomic nervous system is again divided into two branches, sympathetic nervous system and parasympathetic nervous system. Why, ma'am? All are motor neurons, no? Why they have to be divided again into sympathetic nervous system and parasympathetic nervous system? I'll explain it in the next slide, ma. Sir, can we go to next slide, sir? Yeah, last slide for today's session. So here, why it was divided into sympathetic and parasympathetic. So see here, ma. If you see peripheral nervous system along with ANS, what I have described you in the previous slide, along with autonomic nervous system, it also includes cranial nerves and spinal nerves. Cranial nerves and spinal nerves. So here, depending upon which spinal nerves are involved, we divide this autonomic nervous system into sympathetic and parasympathetic. So once you see sympathetic outflow, what is sympathetic nervous system? If the spinal nerves arising from thoracic vertebrae, T1 vertebrae to lumbar, L3 vertebrae, all the spinal nerves from T1 to L3 comes under sympathetic nervous system. That is thoracolumbar. Since the nerves are arising from thoracic and lumbar region, thoracolumbar outflow. Then what is parasympathetic? Parasympathetic mainly includes cranial nerves, that is 3, 7, 9 and 10. 10 is very important, vagus nerve, which play an important role in parasympathetic nervous system, and also sacral nerves. So since it involves both cranial nerves and sacral nerves, we also call it as craniosacral outflow. Right, ma? So here I will take an example of heart. Sir, can we go to homeostasis slide, sir? First.
Yes, madam. Yes,
slide. Yes, sir. Slide number 8, sir. Yeah, can we come back to here, ma? Can we come back to this slide? What is happening to blood pressure here? So I have taken effect of sympathetic and parasympathetic nervous system on heart and blood vessels. I have not taken entire system. So what happens to heart and blood vessels? If sympathetic system is activated, what will be its effects on heart? On heart, it shows increase in heart rate, and on blood vessels, it shows vasodilation, vasoconstriction, both will be there depending upon the type of receptors. So we'll not consider blood vessels, you will be confused. Let us come back to heart. So the effect of sympathetic system on heart will be increase in heart rate. The effect of parasympathetic system on heart will be decrease in heart rate, right? So remember this. We'll move to last slide. Sir, can we move to last slide, sir? Yeah. Yeah, ma. So come back to this diagram. You can see here, I have taken an example of heart. So this red line indicates a parasympathetic nervous system. Ma. I told you vagal nerve, that is cranial nerve. You can see even in the diagram, the red line is starting from the CNS, central nervous system. The nerve is going and the nerve is meeting another neuron at the heart. So how these neurons are classified? Because one nerve alone cannot go from one brain to heart. Usually there will be 2 or 3 in sympathetic nervous system or parasympathetic nervous system. There will be involvement of 2 or 3 nerves, right? So how they are classified? Here the junction between the 2 neurons I have represented with a small dot there. That is called ganglion. What is ganglion? A group of cell bodies is called a group of cell bodies. I didn't say entire neuron. A group of cell bodies of neurons is called ganglion. So the nerve which is before to this ganglion is called preganglionic neuron, and the nerve which is next away from this ganglion is called postganglionic neuron. So the brain, what is ANS? I have simplified it. What is ANS? ANS is nothing but nervous system which deals with motor output. What is motor output? Information is going from brain to other organs. So here you can see the parasympathetic system. The information is going from brain to heart. From brain to heart. So here we have preganglionic neuron and postganglionic neuron. And here we have gap, no? There is a gap here between the neurons, that is preganglionic and postganglionic. How the impulse or information is transmitted through this gap. What is the gap called, ma? What is this gap? Synapse. Very good, very good. The gap is called as synapse, right, ma? So the information is carried from one neuron to another neuron by neurotransmitters. Neurotransmitter, it is very simple, which transmits the information from one neuron to another neuron. So here the neurotransmitter is acetylcholine in parasympathetic system, right, ma? So now what is the effect of parasympathetic system on heart? It reduces the heart rate. So when the sympathetic system is activated, if heart rate is increased, you are in anxious state, you are in a state of fear. So here obviously our heart rate will be very high. For example, any of your staff suddenly comes to the class and say, I will conduct exam today. Obviously you will be in tension, correct? No, right? Or you would have studied another subject during final exams. Suppose today HAP is there, you read PIC. Obviously tension will be there. So immediately parasympathetic system will be activated to reduce the heart rate. Correct, no? So here, fine, ma. So we have not read anything about this. No nervous system, no parasympathetic system. So when you go to 4th sem, in 4th sem, you will have a heading in pharmacology. Parasympathetic drugs or parasympathomimetics. What is this? The name itself is very big. How to study? Very similar, ma. Mimic means what? Mimic means which will act similarly. So here parasympathomimetics means drugs which are acting similarly to acetylcholine. What is acetylcholine here? Acetylcholine is a neurotransmitter. So if you don't read any of these things in HAP, no doubt that you will not understand anything in the pharmacology. So here parasympathomimetics, drugs which act similar to acetylcholine. Parasympatholytics, drugs which act opposite to acetylcholine. So it will be very easy for you if you understand HAP. So similarly, sympathomimetics, here sympathetic system. See the sympathetic system, here also we have preganglionic neuron, postganglionic neuron, and here the neurotransmitter is adrenaline. That is, all the actions are mediated by adrenaline. So adrenaline increases heart rate, adrenaline causes both vasodilation, vasoconstriction. You will read it in ecology. So here sympathomimetics. Sympathomimetics means drugs which act similar to adrenaline or adrenergic drugs. Sympatholytics, drugs which act opposite to adrenaline. So, before the staff explains you, you can come to an idea that what are these drugs, how do they act, right ma? So this is the importance of HAP and any subject, not only HAP. Simply if you read, it won't be helpful ma. So always when we are studying anything, we have to remember three things: understanding, applying, and analyzing. After understanding, you have to apply and analyze it or interpret it. Then only you will be able to understand any subject, right? Fine, ma. So this is the last slide. Sir, can we move to last slide, sir? Thank you for having patience and listening this session for 1.5 hours.
Ma'am, sorry to interrupt, ma'am.
Any questions? Any questions? If anybody wants to ask any question.
Yes, ma'am.
Yes.
Thank you,
Ma'am,
ma'am.
does every drug have the same metabolic rate, ma'am?
I didn't get you, ma. Can you type it? Because voice is not that clear. Which both have same metabolic rate?
Excuse me, madam.
Yes. Yes, sir.
Now, whoever want to interact with madam, please stand up there so that everybody can understand that you have a question, so that all the persons will not activate their voice. Okay. So please, if you have any question right there, you just please stand up so that you can ask the question directly. Others, please mute at that time. Yes, please carry on. If you have any questions, you can just stand up. Anusha madam, please take care of that. Thank you.
Anybody, ma? Anybody can ask any questions. If you have any other questions and other topics also, you can ask me. I'll try to clarify.
Yes, Uday Kumar, you have any question?
But somebody asked about metabolic rate. Somebody, something about metabolic rate you have asked me.
If you have any doubts, students, if you have any doubts, just you get explanation from madam.
Yes, ma?
Ma'am, does same drug has the same metabolic rate, ma'am? Every drug.
No, no, ma. Drugs will not have same metabolic rate. Metabolism varies from drug to drug because under metabolism again we have many reactions like phase 1, phase 2. So what type of system is involved in the breakdown of the drug, depending upon that metabolic rate will differ, and it also changes the excretory rate of drug, excretion of the drug. Generally we say T half, no. So what is T half? Half life of a drug, time taken for the excretion of half of the amount of the drug. That will vary. One drug will have 8 hours, one drug will have 4 hours. So the metabolism varies, and the metabolism doesn't depend only on the drug, it also depends upon our genetics. Few people will be fast metabolizers and few people will be slow metabolizers. It depends upon race of the population, and it also depends upon genes of the population. For example, I'll give you a small example. Some people will be there too lean, but they'll eat a lot. Never their body weight will increase. Correct? No. Few people will be too obese, but they eat very less. Still they'll be obese because that is due to change in the metabolic rate of the body. Clear?
Excuse me, ma'am.
Yes, ma'am.
I want to clarify about pharmacokinetics and pharmacodynamics. I think
Yes,
what does the drug to body means pharmacodynamics. What does that body to drug means pharmacokinetics. But
Yes, ma.
madam told
Kinetics. Yeah. Let me simplify this. Pharmacokinetics means what happens to the drug in our body. What happens to the drug in our body. Pharmacodynamics means what drug does to our body. What drug does to our body. For example, paracetamol reduces our body temperature if you have fever, right? So how it acts, what is its mechanism of action, how it acts, all those we study under dynamics. What drug does to our body, right? Kinetics means what body does to the drug. That is what happens to the drug inside our body. What body does to the drug, right? Like absorption, distribution, metabolism, excretion, all those we deal under pharmacokinetics, right, ma? And here we don't apply only for drugs, ma. We apply this kinetics even for toxic substances. To know about toxicological studies also, we apply this pharmacokinetics. What is the toxic dose level, like that. Clarified, Ganesh? Still you have doubt, Ganesh?
yes, ma'am. Yeah.
Yeah.
Thank you.
Yeah. Yes, sir. I would like to thank once again the management of Shashikant Reddy College of Pharmacy and Dr. Haribhaskar, sir. I know, sir, from the days I have started my career. Thank you once again, sir, and Dr. Praveen Kumar, sir, Vice Principal, Shashikant Reddy College of Pharmacy, for giving me an opportunity to have a wonderful session out there. Thank you once again, sir.
Thank you. Thank you,
Thank you,
Ganesh.
thank you, Aparna. Still you are remembered
No,
with your
sir.
no, with your sincerity and committed work. Still we remembered. Though 12
Yes,
years gone,
yes, sir.
still you are remembered by your the way of you are doing those days. No, you are
Yes,
showing
sir.
lot of commitment, very sincerity. It is
Yes,
going on in my mind from the starting of this seminar, this webinar, till
yes,
now. Yes,
yes,
you explained in a dramatic way.
yes, sir.
Really, what I felt, no, just I'm
Yes,
sitting in the HAP class while I studied 20 years back, 25 years back, you have taken me. Such
yes,
a wonderful webinar, speech you given for my
yes, sir.
students. Yes, of course. It is completely organized by Praveen, Dr. Praveen. You know very well about your classmate for him.
Yes,
Yes, because of him, all the seminars, you can get a feel proud.
yes,
Your classmate doing such a wonderful things in our college. He makes Sasikant Reddy College as alive as possible. So thank you once again for your wonderful speech for 90 minutes, more than 90 minutes, I think. But simply it looks like 5 minutes seminar, webinar. Such a way you taken. I
yes.
think you are working in Karnataka. That's why you forgot a Telugu language, I think.
Sir, no,
Because
sir.
no, you are coming here, Andhra, our
Yes, sir.
students, because it is my just suggestion. You make
Yes,
some Telugu, then students they are heading so closer with you. The speaker who
yes,
is talking the mother language, then
yes,
the psychology of the students and the psychology of the speaker, it is it matches, frequency matches.
yes, sir.
But I thought that you so long years you worked in Karnataka. So you forgot that you are a Telugu woman. Anyhow, thank you very much. Really, for past 100 and so minutes, you are making a very entertainment. From the beginning itself, I told no. After this seminar and after this webinar, I'm sure that our students enriched with some knowledge after completion of your speech. That you makes that is true. I hope the webinar, the attendees, those who attend this webinar, they felt like that only. I hope. We take the feedback also after this session, and then I will send you. Thank you once again
Yes, sir.
for accepting our request and
Yes,
make this evening as a wonderful evening and useful evening for my students. Thank you, Mrs. Aparna Lakshmi. Convey my records to your family and,
yes. Thank you, sir.
and I'm wishing you to get everything for your future endeavors in the field of pharmacy. Thank you, Aparna Madam. Thank you very much.
Thank you, sir. Thank you so much, sir.
Thank you.
Sir, sure, next time I'll take in Telugu, sir. You arrange for next
Uh,
time.
no. That was my suggestion. That's that thing.
Okay, sir. Thank you.
Okay.
Yeah,
Okay, Praveen, sir, you carry on, sir.
yes, sir. Yes.
Yes.
Yeah,
Uh,
thank you very much, madam. Uh, so I have just given a time for you that please, Aparna, can you take for about 1.5 hours? So 2 to 3:30. So
Yes,
somehow
sir.
you managed that exactly by the end of 3:30 you have completed your webinar. So actually the
Yes, sir.
management of your time is very good. So I know you
Thank
from B Pharmacy. Your
Yes, sir.
time management is very nice, so that you got gold medal.
Sir, not like that.
Really,
Casual,
so that
not like
you got
that. Sir.
gold medal and understanding the things by you is very nice.
Yeah.
So thank you very much, madam. Please, I will share you the certificates. Please receive this through online.
Yeah, sir.
Yeah, one
Sure,
second,
sir.
madam. I will share it.
Sir, can you ask students to interact, sir? I hope at least something they understood from the session.
No.
No,
They
no,
are
it's a wonderful session, no doubt at all. I
no
know
doubt at
about my
all.
students. You see, it
Yes,
is
sir.
very difficult for my student to stay there for 90 minutes. It is not that much easy. But you
Yeah.
make it simply, you make them to sit and almost 1.5, 2 hours. So
Yes, sir.
everybody, yes, they are showing a lot of interest. Attendees, I am not only talking about my students. All the attendees, I appreciate them because they are having the patience. They know, they want to know many things.
Yes, sir.
So yes, but your management is extraordinary and really amazing. 90 100 minutes. So you are taken without any slagging. That was important that you do because yes, you are a gold medalist. No, you
Sir,
have the letter. Yes, of course. B Pharm and M Pharm both you are gold medalist. So yes, you proved that one. That's it. You don't want to prove to others also. Just you
yes.
prove yourself. That is enough. So thank you, Panalashmi. Once again, just you get the certificate through online. I
Yes, sir.
request all the students to stand and clap for the time of our Praveen given the certificates.
Thank you, sir. Thank you once again, sir.
Thank you, Aparna. Thank you very much
Thank
for
you, sir.
giving this
Thank you, sir.
great webinar to us.
Thank you, sir. And also, I appreciate the patience of students, sir, for sitting for 1.5 hours and listening this.
Yeah, really, actually,
Yeah.
I am thankful that our students are very patient. That the seminar is like that,
Uh,
that you managed them like that. That's it.
Thank you, sir.
thank you, Aparna. Nice meeting. We'll meet soon.
Yeah, sir. Thank you, sir.
Sure.
Thank you very much, everybody. You can leave the meeting. Thank you.
Sir, can
Sir,
I
vote of
log
thanks.
out?
Just 1 second, sir. I will say the vote of
Uh,
thanks. 5 minutes. Uh, so now I would like to thank first and foremost our management, Sri K. Siskant Reddy sir, and the president, Sri K. Radhika madam, for giving us all the things what we asked for conducting whatever the events that may be the internet, that may be the things which we asked in minutes. Okay, so because this may be a tough time for the management to that to maintain all the things, so but our management is very enthusiastic that they have provided whatever that were asked to us. So thank you, and I hope the same thing will be moving forward in the coming years also. Thank you, the management, for this thing, and I thank Principal Sir for giving this opportunity and providing this opportunity for our students to have this particular very good live webinar. And this is, I can say that this is the first YouTube broadcasting by our college from the Nellore itself in these 13 years. So I can proudly say that. So this is the first broadcasting event from the Nellore live from this center. And I will do my best for that. And this is totally because of you, sir. You gave me the total rights to me, which through that which I can able to do. And coming to the staff, actually, So yes, whatever the things we say, even myself and Harbhaskar sir, so they politely, they don't refuse anything and they will do each and everything what we say. Thank you very much, Shahina, Farida, Anusha, Kaveri, and other non-pharmacist colleagues also too. Very thank you for that. And finally, my students, very thank you for sitting silently for about 1.5 hours really, and helping me all the ways, in all the ways, in all the ways. Not only about the education, not only in the physical work. So each and everything for helping and bringing this institute to the higher standards. So thank you very much, sir. If anything to say, please carry on, sir. Thank you.
No, sir, nothing, sir. It's a wonderful speech.
Yeah, yeah, yeah.
We can wind up.
Thank you, sir. Thank you very much, sir.
Thank you, sir. Thank you very much, sir.